You're listening to DFZRadio.com.
Welcome to A Girl in a Garden on DFZRadio.com. This is your host, Emily. And I was traveling last week. I actually made it back. I was in China all week. And I'm a little tired. And next week, I'll be sharing some of my pictures. And if you would like to see a few of them, I've posted a few on Facebook, facebook.com/agirlinagarden. But I visited a conservatory, some amazing parks, saw some hydroponics growing, an urban farm. So I'll be talking about that all next week. Now, this week, we are going to be talking seed starting indoors and tissue culture. And it's the perfect time to start an indoor garden, especially when you live in the Northeast or Michigan area and you are buried in snow. Tissue culture is an amazing thing. You're taking a genetic copy of a plant on a micro level, very small cuts as opposed to clones. Tissue culture you can do so much with, but it's all about the technique and cleanliness. In the very beginning, you can have a large failure rate, but as you hone in with your technique and have general clean practices while doing tissue culture, you can be extremely successful. So we're going to take a brief break. We're going to hear from A Girl in a Garden show sponsors. And if you are interested in being a sponsor of A Girl in a Garden, please send me an email, agirlinagarden@gmail.com.
You are listening to the beautiful Emily Walter on DFZRadio.com.
DFZRadio.com.
A Girl in a Garden quick tip. Have weeds in your yard? Vinegar is a great alternative to chemical sprays. It's a great burn-down herbicide. Use carefully since it can kill any plant that it touches. For more information and tips, tune in Tuesdays at 5 p.m. on DFZRadio.com.
Here at Green Grow, we take organic gardening seriously. With our mycorrhizae products, you'll increase growth and help reduce the need to replace old, worn-out soil. We help provide electrolytes and maximize nutrient intake to make your indoor garden lush and green. So give your plants the strength that they need and breathe life into your soil with Green Grow. Find us at your local nursery or visit us at thegreengrow.com. That's the green, GRO.com.
You're listening to A Girl in a Garden on DFZRadio.com.
Welcome to A Girl in a Garden on DFZRadio.com. This is your host, Emily. And today I'm talking with Mr. Bill Graham.
How do you do?
I am doing good. And we're going to be talking about tissue culture.
Yes.
So what company are you from?
Pure Food Gardening, San Carlos, California.
And if you do not know Bill Graham, he has been in the industry for many, many years. And he has paved the way for some of this tissue culture that we see here. And you actually have a kit that you're selling too. What is it?
I do. There is the Microclone and Superstars Plant Tissue Culture Kits, which have all the parts in them, except for a few that you'll get locally, and a starter kit. We also make the clone wrap tray, but tissue culture is available as a starter with small tubes I'll show you, and the larger one with extra vessels.
So today it looks like we are going to be tissue culturing some strawberries. So what do we going to do here today?
Okay, well this is the introduction part. This is the piece of plant that we're going to put into tissue culture. I haven't shown you the vessel yet, but we're going to trim these guys up and put them in the tissue culture media, which is sugar, agar, nutrients and hormones. We've made these ahead of time. The plant pieces we're going to start with are some of the fresh growing tips. You know, these are basically pre-flowers, if you can see those.
So these are strawberry plants that we
Yes.
picked from my garden, and we're going to tissue culture them and see what we can grow indoors in hydroponics, basically. So what do we get to do now?
Okay, what I want is I want pieces here that will not be so long that they go through the gel into the bottom of the container. I want them as large as I can because the more tissue here, the more stored energy it has to kind of get used to the new environment. And number 3 will be the smaller it is, the less likely it's going to be surface that's been exposed to mold and mildews. We're trying to do all this stuff clean. There are preservatives in the kit and the media to help it, but we're always treating it like, you know, we're trying to do hospital sterile and let you know, good behavior keep everything clean. Here's 3 pieces. And I'm going to trim these down to the size we want to wash. Put them in a wash jar. The lid and screen are included in the kit. Very popular size jar. I use the 1 pint size. Take these guys, and I only want about 5/8 of an inch below that node. That's going to be the surface of the gel. 1, 2, 3. And so I'm going to take these guys and throw them in here. Put the lid on this, and we're going to move over to the sink. The sequence of cleaning these is going to be rinsing them under running water, adding a drop of dish soap, washing them in soap water, draining and rinsing that, and then I will give the plants in the jar a spray of rubbing alcohol, which I got here. Just 30 seconds washing the alcohol. What I've done is I've used soap and water to wash off dirt and bugs, alcohol to dissolve any oils that might be hiding anything, and then we're going to finish by making a 10% solution of bleach, which I brought here, an ounce of bleach, and a couple drops of dish soap, and that is going to wash here for about 15 minutes before we put it in tissue culture. I'm going to rinse this in the sink. We're going to do a couple of jumps from rinsing it in the sink to mixing up the solutions and spraying it with the alcohol.
And the bleach. Can you tell me a little bit about what the bleach does?
The bleach is going to clean the surface of any mold spores, because we're going to be putting this on a gel that's got sugar in it. We don't want things growing in there besides the plant.
And the bleach is obviously going to kill the fungal and
It's going to kill as much as
bacterial
we can reach.
pathogens that might be on there. So this
Yes.
is a cleaner way. Obviously, for real, like live plants, you don't spray bleach on plants. It will kill them.
Well,
But-
you always want to rinse it off. But
Yes.
there's a lot of peroxide spraying. And we'll do the same kind of thing with peroxide solutions too. Typically, a 20% or so solution of hydrogen peroxide is excellent for rinsing and soaking the plants. I've actually got some plants that I thought were threatened, and I made the 20% peroxide solution, and they are in the cup soaking right now, and I've been gone for almost 30 hours. So I'll rinse that in some sterile water, and which we're also going to use for this process. And let's go ahead and start rinsing these plant seeds. Can you guys see the sink? Yeah. Just shaking and rinsing. I recommend doing this for probably a minute or so. This is the first part of the rinse. We'll speed it up by going ahead and adding a drop of the soap. And at the same time, we're using this to clean the vessel. So as we go through the alcohol and bleach, the inside of this bottle will be sterile too, and then we can move it to the tissue culture cutting area, which we have set up in the other room. Cut. This is like going to be several minutes.
Nice Rolex, Bill.
Oh, it's an awesome Rolex. You know
Okay.
it.
Daytona stainless steel.
That's all right. Are you appreciating the Rolex watch I gave you?
Driving me nuts today.
Gave you a Rolex watch.
Carla.
Okay. So I've been sort of shaking it around in the soapy water. You notice that those are uniform size pieces. I'm just trying to guarantee that I've washed all these surfaces. 5 minutes or 10 minutes or so in the soapy water. There's no real limit. Probably no more than an hour. But the more clean, the better.
Now, does it matter what kind of soap you're using when you're cleaning?
The cleanest soaps you have. Plain Blue Dawn, like they use for cleaning the seabirds after BP.
Okay, so the basic Dawn dish soap, some of the cleanest safer things that safe for wildlife. They
Yeah.
use it for
Probably peppermint oil, Dr.
Bronner's.
Bronner's peppermint oil. Yeah, something like that. Clean soap is really what we're looking for. In fact, now I think about it, maybe Dr. Bronner's isn't great because it's got all the peppermint oil. Anyway, the simpler the better. Here are the pieces all drained. Where we're going now is just to guarantee that I've broken all those oily layers. I like to come in here with a couple pumps of rubbing alcohol, just enough to cover the plants, and toss them around. That's going to be 30 seconds and no more. I want to rinse this, wash it for 30 seconds inside, get in all those little cracks, clean the jar, 15 seconds, and then I'm going to dump this in the sink and fill this with water and my bleach water solution. 30 seconds, good. Rinses the alcohol. Sterile water can be good for this step too. For those of you who have bought the starter kit, the sterile water is included in the kit. It's already been sterilized, bagged. For those of you with the big microclone kit, sterile water is something that you would have made in the pressure cooker pot when you made the meat jars.
Now, can you use RO water with it?
Yeah. In fact, that's fine. When it comes down to the water, the minerals are going to be less important unless it's, heavily hard. It's a hard water. Out of the tap is not bad. We're more interested in microorganisms than the dissolved salts and so on. It's not really nutritional. It's just rinse. It's like car wash water. I guess you do care with a car wash because you don't want the car to be. All right. So that all done. I'm going to put in about 10 ounces of water, and here you go. 1 ounce of bleach. That's going to give me a 10% solution of bleach and water. That's potent. I mean, this is the strength we would use to clean grout at your house. You stay right there. I'm going to add a drop of detergent, and I'm going to run just a little more water in here so I can get to the 10-ounce line. There we go, right up there. 15 minutes. This is when you start setting the alarm on your watch or something like that, or your telephone.
So have a kitchen timer?
You can smell this. Yes, everyone's got a kitchen timer. It's your phone. So use your timers. At the same time, it spills a little, which is why I do it over the sink. But I use that extra water to sort of bleach my hands. And you don't have to shake it constantly. Not a good idea to wear black shirt.
No. So we're going to leave this sit here for about 15 minutes?
Yeah, but I'm going to swirl it once in a while.
Okay.
But, we'll come by and just sort of look after it. And then when I've drained that, we'll carry it upside down and clean with the paper towel on it to the sterile area, because these plants will have gone from being cleaned to the sterile environment of the tissue culture hood.
And this is one of the important reasons why we want to treat our grow rooms, grow areas like a lab. Keep it clean, keep the pathogens out of it. I always like to treat mine like a lab, so clean, clean, clean.
Yeah, sweep the floors, take out the trash, don't let cobwebs build up in the corner. Wipe down all surfaces and run HEPA filters for any garden, not just the tissue culture lab. How do you do? This is Bill Graham again with the actual plant handling part of the tissue culture kit.
So now we are in the grow room, which we have cleaned and made sure we're able to do the tissue culture. And what is the next step now that we've cleaned the plants?
This is where we're going to learn to divide plants, and really this is my chance to show you the vessels and what it is we're really trying to do with tissue culture. We're trying to make many mom plants. This is where your clones are going to come from. If your clones come from it, it's a mom or a donor, and we're going to make small self-contained donors with lots of new propagules, new clone material, growing from a self-contained bottle every 4 weeks or so. Here is a pretty good example. Here, this is a plant that was this large 3 or 4 days ago, but I took this out and divided it into about 6 or 7, even 8 respective new plants from here, and I put one of them into a brand new bottle so that this guy will look like this in 4 weeks. That's the idea of tissue culture. The gel that the plants are growing on is nutrients and vitamins like you're used to. We've got the plant hormone, branching hormone, cytokinins, the opposite of roots. The hormone I've got in here is to keep the plant short, keep it from growing roots, and give it lots of branching sites. We're feeding the plant sugar. So the thing that makes this special is I've added sugar to this nutrient so that it's got a food source, it's not going to have a lot of gas exchange, it's going to grow under, relatively weak light of like 10 watts a square foot, and they're going to produce 5 or 6 times as many plants every 1 month cycle. And we'll do like 50 of these bottles at a time for about $11 worth of ingredients.
And so this one is a California redwood, I think that was done in March.
Yeah.
And this is one that has been sitting under some T5 lighting that I have. And it actually has grown all these different, would it be modules? Or?
Yeah, propagules, new branching parts. How big was that when I gave it to you in March?
I have some pictures of it. I'm going to have to post it. It was tiny.
This
and it
big?
was just one little part of the leaf. And this is really grown just under a T5 light. You don't open it. You keep it sealed. You can see this actually eating some of the sugars, the carbohydrates. It has tap roots that are coming out of it. So this will be split up and made into more again.
And what you'll notice, for instance, is you can see roots growing in this bottle and this bottle. And I've told you that these are not rooting solutions; these are multiplying. And so, why would we have roots? And the reason this has happened is that the plants have gotten big and independent. They're very large, as you can see, and so their own indigenous hormones have kicked in. As the hormone in the bottle that we put there in the beginning is sort of wearing out; it's getting weaker, and so the plant will get big enough and start some roots. This is nice. This is good, because this is one of the factors that tells us where are these plants going to go when I'm harvesting out of this bottle. If we're making 1 into 5 new plants, I only need 1 to make this all over again, and that's the magic of multiplication. But if this turned into that, and I only needed this to get it all over again, I've got 4 new little clone babies, and especially these guys right here that have already started roots, or the root ones here that have already started roots. That's an extra head start. You'll see in the kit there's actually a media ingredient kit, and everything's pre-measured, so you just pour it together with sugar and pH it. But I've got rooting tissue culture media as well, because we'll put small 1-inch pieces on rooting gel so that we can get them up to 2.5 or so and start growing roots. That is an excellent plant to take out of tissue culture. So let's cut some plants.
Let's cut some plants.
Yeah. All right, let me show you what I got here. Cameraman, need a break? What I've got is I've got a smooth plate to work on. We have used rubbing alcohol and sprayed down the inside of the vessel and wiped it all down so my surfaces are clean. Always a good habit to put a little extra spray on bottles that have been closed for a while. And this is the most important part right here, is clean tools. I've got an 8-inch forceps and a simple little stainless steel blade surgical scalpel. Getting a little rusty, but these are in a solution of 70 to 90% alcohol. This is just your regular CVS alcohol in here, and I use the jar to just keep it from tipping over. Now, similarly, I'm going to make sure that all my handles have been treated. An alcohol spray just guarantees that nothing's growing on there that we haven't asked for. Spray my hands. And before I get going too fast, Emily, I should probably ask if you have any questions at this point.
I think I'm going good right now. Let's see you cut some plants up.
Okay, so I'm going to take the tool out, and I'm going to get this guy. I'm going to go ahead and play with the redwood you made. Before I get too far ahead of myself, I'm going to clean the plate with a few sprays of alcohol and a wipe with the paper towel. Just want to clean the surface of that tray. I'll hold on to this because this will help me wipe plant debris into the trash can. Oh, and I'll need a trash can in the next couple minutes.
We will get you one of those.
Alrighty. So what I'm doing here is I want to take the tools, take the plants out. I want to handle it as little as possible. There we go. Right in the middle of the plate. Isn't that amazing? That was one piece of plant about this big, and this is what it's grown into. All those little plants growing left and right are going to be brand new plants. So this I'm just going to go here and find some little natural fissures, places where it wants to fall apart by itself. I don't need that root, so I'm going to cut it off because I'm not rooting these. And I'm trying to make this into relatively uniform pieces of about an inch or so. So I'm right-handed. This next part will help me move the plants. But I'm done with the scalpel. And I'm going to locate these guys so that the tops are pointing to the right-hand side because I'm right-handed. There we go. Always dip tools. And in this case, I'm going to take one of my screw top tubes. These are included in the kit and in the starter kit. And I'll just grab a plant, tilt the jar, and it goes right into the gel. Oops. Goes right into the gel very simply.
Now, is this gel solution the stuff that's already mixed up?
In a starter kit, these are ready to go. This is a rack of tubes all ready to go. See, the gel's already hard. They're sterile. They'll come wrapped in plastic, so they're sealed. And this is what a starter kit looks like. What we'll do, I do want to move those plants before I wait, but a starter kit has the tubes, the tools, and the jar for washing the plants like we just did in that last video. So what we want to do is you always want to work fast. You know, don't leave your work out on the table if you can help it. I'm going to grab this guy so I'm pretty close to the bottom. When I tilt it, I want to spill out that little extra water, see what I'm doing, go straight for the middle of the gel, and it only takes about a 1/4 inch or so, not even, and it's well seated right there. Perfect. And I still got 2 more plant pieces. I'll use another tube on these. Just good habit to dip the tools between all these handlings, and getting plants into and out of bottles you want to do as quickly as possible. What do you all think? Can you sort of tell, you know, what size the plants are and what we're making here? You see, we're getting new tubes. I've got a choice of what bottle I want to put it in. So a large piece I'll put in a taller vial. These are 6-ounce polypropylene vials with flip tops. We've got really good pricing on these, so just give us a shout. They also fit perfectly 50 to our 10 by 20 skeleton trays. But that's sort of the basics of the tissue culture. I'll clean that plate, and we're really just going to go do all of this again. I'll spray the plate with alcohol and go through cutting the plants.
And now we're going to do our strawberry solution. Well, the strawberries that we're soaking in the solution, those
Yes.
are done now. The
Okay.
timer just went off, so we're going to take that solution
Excellent.
and put the strawberries, well, the cuttings, into
tubes.
tubes.
We'll be doing, you know, really the cleaning you just saw in the jar and the planting we'll do sticking the pieces in here is exactly what the starter kit's all about. This is just a way, without having to do any of your own cooking, these store and ship well. Have tubes of prepared media. Once you put plants into these pieces are all usable with the full tissue culture kits, which are about $253, but there's several months worth of laboratory tissue culture. This guy, all these pieces can be used in the full kit later, so it's natural to start here, see some growth, or learn some lessons at least, and either buy replacement tubes, which are always for sale, or jump up into the full kit where it's a much better value. You know, these guys cost you almost $8 each or so, because they're prepared in the way that everything comes in the kit. You get a full kit, and because it's 50 bottles times 4 crops, that's about $1 each.
So why do people really want to use tissue culture? Why is it so great?
Tissue culture is another tool for the toolbox. Anybody who's cloning plants is inevitably going to run into issues of efficiency of space and light and the time that you're going to put into it. These are questions that all got answered years ago by the horticulture industry and the greenhouse industry. It was just not practical to do standard cloning for crops that you're trying to make 10, 25, 35,000 a day of. And this is just natural. So the laboratories that do this look genuinely like libraries with the narrow shelves, except there are fluorescent lights on those shelves, and the bottles you see here are racked up underneath them. And they'll pull one of those bays and produce 7 bays worth of plants out of it, and then only make 1 more original bay to put back in. 4 weeks later, 7 new plants. And that's why, you know, the commercial horticulture uses this for orchids, phalaenopsis, dracaena, syngoniums and spath and ferns, tons and tons of branching plants and other carnivorous plants. It's very easy, and the kit is excellent for somebody who has the curiosity of this. You've read about it, you've seen it online. Where I stand is about 20 years ago, you had hydroponics and you had hydroponic stores, but people still didn't really know what it was they were looking at. They'd walk into a store and you really were talking them into convincing them that those lights would grow plants. These lights hung around your room will grow plants. Well, now we know that's just a given. We're way past all of that, and where I stand is I am just like grow lights and hydroponics were, but with propagation 20 years later. This will happen, and people will improve on what we've done here. But my idea is to make it fantastically simple.
And this is a way you can also save genetics. Even, well, you're cleaning it and making it sterile, but there has been genetics that has gotten fungal, bacterial diseases on it, and you can use tissue culture to hopefully clean it up, I've heard.
Usually. What you're trying to do is, most of the time, those diseases aren't inside the plant anyway. I mean, maybe inside some tissues like the leaves, powdery mildew would get underneath the cuticle, but it's not traveling in the vessels.
Right.
People get worried about that, but it's not really happening like that. Plants that lose vigor over time, you know, especially from repeated cycles of cloning, usually they've got a pretty cushy environment. I mean, these plants are very happy in the environment they have. What we want to do is kind of wake up genes that are asleep. You know, DNA doesn't change, but genes can turn on and off. It's going to be turning on and off the right combination of genes and how much. And that's what we're always trying to do with the plants. Tissue culture is just another tool for doing that, but I actually recommend your listeners, make solutions of Super Thrive or some of the Thrive products, and do foliar treatment of those before they get into tissue culture for vigor revival.
Okay, so let's do the strawberries.
All right.
Okay, so now we have the strawberries, and that's been in the bleach solution, which soaks for about 15 minutes, and now we're ready to rinse it.
You got it. We're going to just drain it. I'm going to drain this, to save it, because we'll use the bleach water to wash down the hood. But now that's drained, I'm going to carry it upside down with the paper towel to sort of protect the ground. Mold spores don't fall up, so this is safe. And we're going to get this to the hood, where we're going to pour sterile water over it to start diluting the bleach and rinsing it away.
All right, let's do this.
Two.
All right, so welcome back to A Girl in a Garden on dfcradio.com. Now that we have the strawberries that are clean, now we're going to put these in tissue culture. So what do we do, Bill?
All right, so we drain the bleach water off of these guys, but they are now still covered in bleach. And what we're going to do is, I've got sterile water here. This is water I made ahead of time in a mason jar. Tear into this sucker, and I'll pour a third of it right in through the screen to rinse these plants. So again, it's a sterile hood, so I want to do all these in the protected area. Oh, isn't that awesome? That's the seal of sterile water. Used to be the lid to a pasta jar, as you can tell. Screw that back on and leave it here. Kind of uneventful. I'm going to leave these soaking for 1 to 5 minutes or so, and we're going to do that like 3 times.
1.
We've sort of finished our first soak. Doesn't look like much. I mean, these guys are just floating in the sterilized water solution. That's just a jar of water that went into the pressure cooker at the same time that these were produced. You'll get very used to using the pressure cooker and preparing all these things. See the other videos at Microclone on YouTube. But I'm going to go ahead and drain this out and replace it with a little more sterile water. And it's really just that simple. Drain it, shake it down.
Now, can they use distilled water?
For this, since it's rinse water, the salt content really only makes a difference if it's extremely hard water, difficultly hard water, because that could affect its ability to rinse properly, as it wants to combine with soap. But You know, if you're below 200 or so like that, then tap water is just fine. But again, to sterilize it. Where distilled water is good is when we're preparing this gel that the plants are going to grow in. There, distilled RO is more important.
Yeah, and this water, actually, I'm out in Northern California. I think it's like 70 ppm.
70 here?
But it comes out of the faucet, so it's pretty
That's
clean.
actually great.
Yeah.
You're in good shape. Here you go. Everything I spill, I use as an opportunity. I move the paper towel in here, and it's always good to be cleaning the area here. I want my hands to be clean. I don't want to reach over any plant materials, so I hand myself tools. You know, we want to be able to move in here without draining anything. So I'm going to go ahead and start taking that first plant and show you how to put it in a tube. 3 sprays.
And it's nice to have your workstation all set up so everything's right at your fingertips. So you don't have to keep getting up and grabbing other stuff.
Absolutely.
And that can cause cross-contamination too when you're
course.
getting up and grabbing stuff. So everything at your fingertips, get organized beforehand. It makes it much easier.
There you go. I'm going to just drain out some of this water I don't need. And I'm going to set this on its side, again, because spolars don't fall sideways. And just very simply, one at a time, take this little fella out, and use my tools. I want to trim it so that that is just about as deep as the depth of the gel. And then we'll just plant it right on into the one of the gel bottles. This is only about 20 milliliters of gel in this bottle. I lift the handle up, and in it goes, right there. Best part about this placement is the node, right where the plants are going to grow from, that growing point, is right where the gel meets the air. That's going to be the area heaviest feeding. Tissue culture plants don't draw their nutrients up through the bottom because they don't have the roots. The vessels aren't really transporting because we've lost transpiration, where water usually will leave the surface of the leaf and new water comes to the roots. We have a high humidity, so that doesn't happen. This is going to be direct absorption. So the first thing that happens this first month is the plant learns to absorb its sugar and nutrients right through the skin at the point where it's in contact with those growing points. Now, Carlos went with me to Epcot Center, and that's one of the great places to see a tissue culture display because they've got the greenhouse with all the hydroponics and the land exhibit, and this is what they've been famous for since Epcot opened. And one of the highlights of the hydroponics exhibit is the tissue culture lab with thousands of jars and tissue culture plants just like you're looking at in my hand for sale in the gift shop and down at the doors to the exhibit. So it's very simple. There's purposes in doing it. If you've ever been interested, if you've seen it at Epcot Center, you've ever been interested in tissue culture cloning, you're doing any kind of cloning right now, or you even have plants that could value from this, like violets, carnivorous plants, ferns, or what have you, valuable trees, then don't hesitate to purchase the kit, source a large pressure cooker about so big for sterilizing everything, and then just give yourself a clean place to do your work and a lighted, clean area to put these plants to grow. Besides the stuff we do in the kitchen, right at the stove, there's some stove work. You're looking at what we're doing in the hood here and then putting them on a shelf to grow. That's tissue culture. You can do all of this in a 4x4 hut and have plenty of room.
And I will say Bill is an expert at this, and he has been doing this for years. So be prepared to give it the college try more than once. He's making it look really easy, but it's a fun thing. It's good to experiment. It's a cool science project. It's fun to do with kids too, if you want. So
excellent, absolutely. I have shared these materials with middle school students, given the plants as gifts. You know, my garden now sits on a shelf and resides in these little bottles. This is a great opportunity to be able to create a garden that's easily managed because it's right under your nose. It's right in front of you all the time.
And I will say Bill Graham is known for having his California redwood, especially with us being from California. But I ran into him in other countries even. It was in Barcelona, and
Yep.
he has tissue culture plants with them.
That's right. These guys travel so well that what you're looking at right here, these bottles, have traveled with me to many places. Spain and Europe, Vienna, the UK, Holland, France, Germany. It's too easy. I can just, what you're looking at right there, we will take through customs. And we go through the airport screening just fine because there's almost no liquid. And at customs, you want to be truthful, but in Europe, they don't tend to ask about the live plants. It's everything else. So I've yet to declare them, although a lab can have a certification provided that the material leaving that lab is phyto sterile.
Mm-hmm.
Same kind of thing. I'll always have to twist my arm, but each one of these guys is going right in the middle like that. Give it a little drain. That is how, my friends, we're going to start new plants. There they are, 1, 2, 3, brand new strawberries right there.
So here we go. We have strawberries. I have a feeling I might bring these strawberries to the Maximum Yield Indoor Gardening Expo next month.
Ooh, that would be fantastic.
So stop by. I'll let you guys know which booth I'll be working in, but you'll have to stop by and check out some of this tissue culture that we worked on. Obviously, some of these are from March. We just split California redwoods. We're going to have the brand new strawberries. So I'll kind of give a progress update on A Girl in a Garden on Facebook, just to see how these are going, and then you can check them out in person if you're going to be at the San Francisco Indoor Gardening Expo. And that is July 27th and 28th, so coming up pretty soon.
28th is public day.
28th is public day. So if you are the general public, come on Sunday. So I have to say thank you very much, Bill,
My pleasure. Thank you
for
for having
showing me more about the tissue culture. Hopefully we can shed some light on tissue culture. And This is Emily from A Girl in a Garden on dfcradio.com.
It's been my pleasure.
You are listening to the beautiful Emily Walter on dfcradio.com.
dfcradio.com.
A Girl in a Garden quick tip. To keep hungry insects and slugs away from your cucumbers and vegetables, mix strips of aluminum foil in with your garden mulch. As a bonus benefit, the foil will reflect light back up onto your plants. For more tips and information, tune into A Girl in a Garden, 5 p.m. Tuesdays on dfcradio.com.
Here at Green Grow, we take organic gardening seriously. With our mycorrhizae products, you'll increase growth and help reduce the need to replace old, worn-out soil. We help provide electrolytes and maximize nutrient intake to make your indoor garden lush and green. So give your plants the strength that they need and breathe life into your soil with Green Grow. Find us at your local nursery or visit us at thegreengrow.com. That's the green, GRO.com.
You're listening to A Girl in a Garden on dfcradio.com.
dfcradio.com.
Welcome to Girl in the Garden on dfcradio.com. And today I'm doing some seeds and wanted to share some tips that I had with it as well. So what I have for is I have my seeds. I have a little to my potting soil, which I have a premium organic soil that I'll be using, and then I have trays to plant seeds. Productions right here, and I have a marking pen, plants, which are always and so what I'm going to be doing with this is pre-wet I'm doing this, or I'm pre-wet the soil. I'm doing this inside. I don't want to make it. So I'm going to fill up all the cells. When I say I don't want to make a huge mess, I mean I'm still going to. That's what happens when you garden. You learn to vacuum a lot more than you do, and go down a little bit, which is because this is just will be transplanted somewhere else. Actually, does not take either. What I go to water actually in a crock tray, so I soak the method where I can, or I can soak from below, or I can top feed. I typically like feed from the reservoir. That way, it's easier that way. You don't different ways. Season. There's just quite a few different cells that I can start to. There's ones that are 18 size. There's one that are 30 size. There's another one that there's many different containers you can use. Container if you want. If you just in the side, but I'm trying to start this under some lights inside for indoors. So I have my, and then I will take my pen, punch just a little one. Use a pen with your finger if you want the other way. So I just kind of put my pen in. And then when it comes to starting, so pretty large. And once you start seeing or in the garden, you'll start collecting. You will. So I start seeds. I want to start and can't start it indoors. So if you do both items, carrots, radish, something pack root, do not. And I made the mistake. They just don't. Do whole carrot crop, and you're going to do it in the same container. Don't wait. Rex out in a container transplanted. A little bit larger. I think I'm going to do peppers with these. I have. So I have peppers. Get in the flow of it. It's pretty. But I'm going to do cucumbers. there. I find my takes often. What the heck is my plant? Which kind of, and then you start to learn that you must take everything. I don't mind the chaos. I am not someone who chaos. I have friends, and you might type of gardener. Anything grows, it's okay. It's not that plant grow organically. From last year, I'm starting with some new plots. I will be. I like to plan out what space I have. I have some trellis going. Cucumbers are perfect. So I am going to do cucumbers. So just drop them in. Cover them up. Need a little bit more soil, which is right here. White cucumber. And some of these, if I follow them, because they'll go to the local community. And you never know, everything might. So I usually do one seed, these seeds, so they. If one doesn't come up, it's not the end of the world. But starting more than one. So I will continue planting this whole row, and honestly admit I didn't really pick out what I wanted to plant. I need to plant. These are a little bit larger, so I will plant some things. I have some amaranth, kale, one. Yeah, I have. So let's. I also. I definitely want to grow some of that. I'm installing a raised bed. These are fun mix of flour. Also, addition to salad. Rainbow. Here's some more turkey. These are vegetables. They need to be direct salad only. And I have. So it looks like I picked out a few little things. A few seeds. The master show. I'm going to drop seeds in each one. These come out. Now, if you aren't sure how old your stock is, you can plant a few more than needed. And I don't have to plant all of them. These were from last year, so I know everything's. Even with the seed, have a few that's. You're going to have a few. So my. Looks like I just. So for this one's in, I'm going to add a little bit more oil. Thing about gardening seeds and trying items is, but there will be, and typically if you're gardening with your family or kids, they'll be more active. I don't like when they grow it. She might like it a lot. They know. So let's see. We're going to pack. These are packed for 2012. We're going to do 2 seeds each. I want to make sure those come up, and the seeds get older. I probably haven't followed all the seeds for so these are sugar. This is what you see. You get more than you will ever use. You'll give them, you'll keep using them. Some of them might not all germinate, like earlier, that's in the world. I would say from this, I really saving a lot more of my own. I have to buy all these every year. So and I will be seed saving tech. So just a pretty big owner around us. Every single company magazine that you have dreamed of, it can't stay. Now, if you don't have space, these can be indoors under. If you don't have space, it can be. It's really going to depend on your weather. If you live in an area that's getting snow, not going to be very significant until the ground that, or you're going to have a sunny, a little bit warmer. Or it's pretty. You can also get heat. There's light. Kinds of ways that so it's easier for me. Let's see, start something new. Amaranth. These are really seed. They're tiny, so I'm going to wait on this one. Some switch. Seeds very similar. Longer come up. This is a leafy. It's high in folic acid and stuff. There's tons of great chard you have not tried. It can be sauteed, you can eat it raw. If you want, there's all kinds of things. There we go. 2 the chard. And any once the package for the seed package, you won't put it in. Contaminate that way. And I'm just doing vegetable on a small scale. I am not super of it, but you're doing it more on a if seeds or soil is dry, that is considered, and you start with something fresh. We're going to do amaranth. By mistake last year, and it ended up being beautiful. So I used elephant head. These were packed 14. People too, you can seed markers, cut off in a small. I like to use an indelible. Stuff wears off pretty fast. And just put a few of these are tiny, so I'm one in each one. This is to similar to, no, I'm not thinking of for you. They are, well, the ones I saw, and they look like plants with flowers on the top have tons of seeds. That's the part that you're eating. Chia seeds too. That's what I was thinking. Chia seeds. So these in one. So I just planted in just a few minutes, so that's not that hard. Now the next what I do for this all these water. So I'm going to water these from the bottom of this tray. Water. Okay, so I have filtered water that I got out of I'm actually just on the bottom the soil so water from up. I will have to fill this up a few times to make sure this, but I will spread the bar in so it gets nice and wet, and I will be moving plants. So these are going to be grown in my kitchen, and once they're soaked in, they'll be moved into the garden. And what I have for that is actually I can use things correctly. So we have a garden. This is a tent, actually tall, and so this is in my living room. I have some lights installed. I have some plants that actually grow right now. Add these in here, and they will be right now on a timer, so it'll be about 18 hours. Will be sprayed pretty often. I give them some give them a little bit of food so they can get stronger, and time to transit, they'll be transited into a little bit larger, and then when it's time for them to go outside, they will be part of them off. I'll take them outside for a few hours, for a few days. Especially, I am in North or by the ocean. We have the fire that comes. It can be 70 freezing at night. I take my delicacy directly from outside, or I'm inside. I'll have them die. There can be some trouble with them, and it's going to be too cold for them. I'm just going to fry. As well, they aren't going to fry. Excuse me. They're just going to shrivel up. They're going to fry from. So I am going to grow these until they are healthy and large as possible before I send them outside. Healthy, robust seed start before you put them outside. Make sure they're hardened off. Now, if you don't have time to home, you can always plant your local, and I always suggest you can get plants or things that work specifically in the area. I was even today, and they had plant starts that were so they had local Sebastian plant starts. That's pretty common for a lot of your centers too. Is native ones and you can look for ones that were started. Buy those plant starts. Look for something local. I'm not going to buy any. I know I'm lying to myself. Go to the garden. Amazing. What happens? I like. But there's tons of ways to plant seeds. So I will be moving my tray into here in up a few weeks. These seedlings are came in. Emily with The Girl in a Garden on DFCRadio.com.
These are the words of my master.
Welcome back to A Girl in a Garden on dfcradio.com. This is your host, Emily. Big thank you to Bill Graham from Microclone. Thank you for taking the time to talk about tissue culture. Next week, I'm going to be talking about gardens that I visited in Hong Kong. I have guest Sam from Cabinet Grow. We'll be talking about growing in small spaces, cabinets to be precise. And also, I visited Brian up at Vital Garden Supply, and they have a pretty awesome tropical garden up in Nevada City. High elevation, and you can still grow plants year round. So until next week, this is Emily on A Girl in a Garden on Gmail. What did I just say? See, I'm still tired. This is Emily on A Girl in a Garden on dfcradio.com.
You're listening to DFZRadio.com.